Hello students, Jai Hind, Dr. Manisha Patel from Divine Science Academy. In chapter number 2 today we'll see megasporogenesis. This is most important topic. So first of all, what is megasporogenesis? To remember that process of formation of megaspores from the megaspore mother cell is known as a megasporogenesis. So we have seen the structure of ovule. Ovule, it is surrounded by two membranes, two integuments. This is outer integuments and this is inner integument. Integument is surrounded the nucellus. Nucellus possess so many cells. Towards micropyle end, one cell becomes large, contains dense cytoplasm and nucleus, which is known as a megaspore mother cell. Remember, all cells of nucellus are diploid by nature. That one cell which becomes active and which is known as a megaspore, which undergoes meiotic division and forms 4 megaspore. So this is meiotic division. This is megaspore mother cell which is diploid. This is meiotic division and its first phase. This is meiosis 1. Due to meiosis 2, we are having 4 haploid megaspores. Remember, from one megaspore mother cell, we are having 4 cells. But all becomes haploid because this is meiotic division and this is reductional division. So that number of chromosome becomes half. So due to meiotic division from one megaspore mother cell, we are having 4 megaspore. Out of this 4 megaspore, only one becomes active. Remaining 3 degenerate. Only one becomes active and it forms female gametophyte. Actually, female gametophyte, it's a structure which is actually involved in fertilization. Female gametophyte, or which can see it as an embryo sac. In embryo sac, there are 8 cells involved. We'll see in next point. So remember that from one megaspore mother cell, we are having 4 megaspore, but only one becomes active. While from one polar mother cells, we are having 4 pollen grains. This is involved, or this is important point of view for sums. So from one polar mother cells, we are having 4 pollen grains, and from one megaspore mother cells, only one active megaspore we are having. That pollen mother cell, it is also known as a microspore mother cell. In female gametophyte, which is also known as the embryo sac. So how that 8 cells are formed? So according to Panchanan Maheshwari, if out of 4 megaspore, one becomes active and divide and it forms embryo sac, such a development is known as a monosporic development. Why monosporic? Because you can see here, this is one megaspore, it divides and forms 8 cells. So due to first division having 2 cells, this is mitotic division, remember. Due to second division 4, and due to third division we are having 8 cells. So from one megaspore, after 3 division, we are having 8 cells. So such a development is known as a monosporic development. Now if 2 megaspore involved for the formation of embryo sac. So 2 megaspores, so due to first division we are having 4, and due to second division we are having 8. So from 2 megaspore, after 2 division, we are having 8 cells. So such a development is known as a bisporic development. So remember that why 8 cells? Because that female gametophyte, or we can say embryo sac, is having 8 nuclei. So its main target is to form 8 cell for the formation of female gametophyte. And if 4 megaspore becomes active and it forms embryo sac, that is 8 cell, such a development is known as a tetrasporic development. Now you can simply understand that from one cell, if 8 cells are produced, then there are 3 mitotic division involved. If from 2 megaspore 8 cells are produced, then there are 2 divisions, and if 4 megaspores are involved, then we are having only 1 division. So according to Panchanan Maheshwari, these developments are known as either monosporic development or bisporic development or tetrasporic development. Now, actually how this embryo sac is formed. So we have already seen that nucellus, its functional megaspore divides mitotically to form 2 nuclei. So due to first division, first 2 nuclei are formed. Right, this is 2 nucleate embryo sac, again divide and 4 nucleate and again divide, and it forms 8 nuclei. And this 8 nuclear stage, it forms embryo sac. So what is different here in formation of 8 cells? So for the formation of 8 nuclei, there is mitotic division. But these mitotic division are free nuclear. Now what's the meaning of free nuclear division? So remember here, the free nuclear division means that nuclear division occurs, but they are not followed immediately by cell wall formation. मतलब नॉर्मली सिर्फ न्यूक्लियर डिवीजन होता है, उसके सराउंडिंग जो सेल वॉल फॉर्म होनी चाहिए वो फॉर्म नहीं होती है. Right? So remember, this is free nuclear division. That means nuclear division occurs, but it is not followed by cell wall formation. मतलब cell wall form नहीं होती है. So this 8 cell nuclei organize and they form female gametophyte, right? Or which is also known as the embryo sac. So out of this 8 nuclei, 6 nuclei are surrounded by cell walls and they organize into perfect cell. So you can see here, these are 1, 2 and 3, 4, 5, 6. तो दिस 6 न्यूक्लियाई जिसके सराउंडिंग में सेल वॉल होती है, राइट, दे आर अरेंज here and here. That is, you can see here, this is micropyland and this is chalazoland. But 2 nuclei, this one, they are known as a polar nuclei because they fuse together and they forms central cell. So simply this is 8 cell arrangement, 3 toward this side, 3 toward this side and 2 in center. Now exactly the arrangement of embryo sac. Most important in structure of embryo sac, 3 cells arranged towards chalazoland. You can see, this is chalazoland. And they forms antipodal cells. It is located towards chalazoland. All 3 cells are haploid. So there are 3 cells, 1, 2 and 3. They are located towards chalazoland and they are known as antipodal cells. In center, we have learned here, it is polar nuclei, but 2 nuclei. So that's why I have written N plus N. And towards micropyle and there are 3 cells. In center, that is egg cell, which is obviously haploid, and 2 synergid cell. So these 3 cells, that is one egg cell, the center one egg cell, and these 2 synergid cells, they forms egg apparatus. So this is most important structure of embryo sac. One more time, in embryo sac, 8 cells are involved. 3 cells are located towards chalazal end. 3 cells are located toward micropyle, and 2 cells in center, 2 nuclei in center forms polar nuclei. तो egg apertures बोला ना अभी. In egg aperture, it involves one egg cell and 2 synergid cells. The 2 synergid cells, they are having special cellular thickening at the micropyle tip. अब जैसे ये माइक्रोपाइल एंड होगा तो यहाँ पे कुछ पोर्शन स्वोलन होगा, एंड दे फॉर्म्स दैट फिलीफॉर्म apparatus. तो फिलीफॉर्म apparatus is observed in synergid cell. What is the importance of this filiform apparatus? Filiform apparatus helps for the entry of male gamete by pollen tube. मतलब कि जब भी पोलन ट्यूब आएगी तो पोलन ट्यूब यहाँ से फिलीफॉर्म apparatus के थ्रू अंदर एंटर होगी. दैट फिलीफॉर्म apparatus गाइड्स द पोलन ट्यूब. रिमेंबर? ओके. सो मोस्ट इम्पॉर्टेंट, वन मोर टाइम, in embryo sac, 3 cells, they form or they are located towards chalazal end. They forms antipodal cells. 2 central nuclei forms polar nuclei, and 3 cells towards micropyle end forms egg apparatus. In egg apparatus, 1 egg cell and 2 synergid cell. This is egg cell and 2 synergid cell. Synergid cell possess filiform apparatus, just like projection like structure. Filiform apparatus. So the typical angiosperm embryo sac at maturity, most important, it is 8 nucleate but 7 celled condition. 8 nucleate and 7 cell, because this is 8 nuclei 8 है, 1 2 3 4 5 6 7 8 nuclei 8, but cell 1 2 3, this is 4 because it is fused, this is 5 6 and 7. So remember, typical angiosperm embryo sac is 8 nucleated and 7 cell condition. This is most important question. Development of embryo sac, that figure of embryo sac. Remember what is micropyle end, what is chalazal end, number of antipodal cells, all are haploid, how egg apparatus is formed, egg cell, 2 synergid cells, right? What is the function of filiform apparatus? This is most important. And second thing, remember, all cells of embryo sac are haploid. Antipodal cell is haploid, polar nuclei n plus n, egg cell again haploid, even synergid cell is also haploid. Okay, thank you.